The Centre for Membrane Interactions and Dynamics, CIMIAD, is a university centre which includes cell biologists, physicists, chemists, mathematical biologists and clinicians. Our driving force is really to harness our understanding of biological processes to develop better treatments for disease. Cells are the basic building blocks of life. We're all made of millions of cells, and cells have a barrier which protects them from the outside world, and this is called the plasma membrane. And our aim is really to understand how membranes cause cells to function.
You can imagine that the cells that make up your brain and are involved in thought processes are going to be very different to those cells that are part of your immune system, whose job it is, for example, to fight off, say, a virus invading your body.
Within cells, there are lots of different subcompartments which are specialized for particular tasks, like the rooms in your house, where different activities occur in different places. Cells are always taking up material from their environment. This material, which can be nutrients or signals, is recognized by proteins on the cell surface, the plasma membrane. The material is packaged into spherical transporters called vesicles that pinch off from the plasma membrane and enter the cell. Once inside the cell, the vesicles move along protein tracks that are part of the inner skeleton of the cell, the cytoskeleton, and then deliver their cargo into an intracellular compartment called an endosome. The endosome is a major sorting station where the cargo is organised into other transport vesicles to be sent to other compartments within the cell, again using the protein tracks of the cytoskeleton.
Exocytosis is the process used by cells to transport material to the cell surface. There are two forms of exocytosis that take place in cells. Constitutive exocytosis occurs in all cell types, and it goes on all the time. As soon as a vesicle is loaded up with cargo and ready to go, it travels to the surface and then fuses with the plasma membrane to release its cargo to the outside. The nature of the cargo transported through the constitutive exocytosis varies. It can simply be garbage the cell wants rid of, or it may be something more specific that functions outside of the cell. Some cells, like neurons and immune cells, have an additional form of regulated exocytosis. A defining feature of regulated exocytosis is that after the vesicles are loaded up with cargo and travel to the plasma membrane, they have to wait for a signal before they can undergo fusion and release their cargo. Mast cells, the so-called sentinels of the immune system, use exocytosis to secrete chemicals which help fight off any baddies trying to invade our bodies. They also use exocytosis to secrete chemical messengers which call in reinforcements to help fight off the invaders.
As a physicist, my goal is to develop optical techniques to improve the microscopes that are used by the biologists. Lots of the microscopes we have are very good, but they're limited by some of the basic laws of physics. So we have to find ways around those laws. A little bit of cheating maybe. And by doing that we can improve the resolution, and so we can see smaller and smaller features and learn more about what goes on inside the cells. We work quite closely with the biologists to learn about what size scales they want to see, how many different things they want to see in one go, and more importantly how fast they want to see the processes occur. We can give up on some things to gain from other things. So we could give up on resolution and gain in speed, or we could give up on the number of things that we want to see moving and have faster, high resolution microscopes. So it's very important to work with biologists.
So a good example of where exocytosis can go wrong would be in the case of asthma. So in asthma, the mast cells that are living in your lungs respond inappropriately to something that isn't dangerous at all, such as pollen. And what they will do is when the pollen is detected by the mast cells, they will secrete those mediators that are trying to attack the pollen, even though it's completely harmless, and instead those mediators can start to damage the surrounding cells, your own bits of your lung.
I see patients and I see all the problems I can't cure. And this is what sends doctors like me to the laboratory, because we know we can't make people better with the drugs that we've got at the moment. We know that asthma is a nasty disease when your body's own immune system turns on your own lungs. And we know that what happens in the lung is that inflammatory chemicals called cytokines are made that cause the lung to become inflamed, make the muscle around the airways twitch and grow so the airways squeeze tight at minimal stimuli, making people wheezy and uncomfortable. So we have to understand what's happening in patients that means they've got disease. And then when we know what's happening, whether we can block it and make new drugs, and we can test those new drugs in the lab and then try and take them back to the patient.
As we investigate more, we realise how much more we need to understand. But we feel that we are making significant progress for diseases such as asthma and Parkinson's disease by understanding membrane dynamics within individual cells.